Hello and welcome everybody to today's Flash Talk. This, I can't believe it, will be the last Flash Talk of the year 2024. The year has been incredibly fast. I'm your host, Dennis Koester, Director of Industry Research and Relations here at Drug Hunter, and I'm joined today by Rebecca Paul from Isomorphic Labs, and we're very happy to have you today, Rebecca, and we're looking forward to your presentation.
Thanks, Dennis. Yeah, it's exciting to be here.
I'm going to properly introduce Rebecca now. So Rebecca Paul is the head of medicinal drug design at Isomorphic Labs, and she holds a MA in natural sciences from the University of Cambridge and a PhD from Imperial College in London. After her PhD, she moved to Heptares Therapeutics, which is now Nxera Pharma, and she's working in the space of GPCRs across a range of different modalities and disease indications. Her first experience working in AI drug design was at Benevolent AI, where she led projects in the neurodegeneration field. And Rebecca joined Isomorphic Labs in 2022, and again, she leads the medicinal drug design group there. And to date, Rebecca contributed to 6 projects that reached the candidate nomination stage or beyond. And there's going to be many more projects that she'll be bringing to the clinic, as we'll see today. So we're very happy to have you today, Rebecca. Welcome to the show.
Thanks very much, Dennis. Yeah, thank you for the kind introduction.
Yeah, absolutely. And we have a excuse me, we have a tradition here at Drug Hunter to do some ice-breaking questions before we start with the presentation to get to know our speaker a little bit more. So, could you tell us a little bit more about what inspired you to start a career in drug discovery, Rebecca?
Yeah, so actually, when I was younger, like a teenager and thinking about university, I actually just really loved maths and physics. And all I wanted was to work for NASA. And no human on earth was going to convince me of anything else. But then I got into Cambridge to do natural sciences, and I ended up taking chemistry as one of the subjects. Totally fell in love with chemistry, especially synthetic organic chemistry, supervised by some really inspirational chemists, such as Stuart Warren. And then in my 4th year undergrad project, I ended up working in Chris Abel's lab. Some really inspirational postdocs working in the lab at the same time as me. So it was a really amazing place to work. That was my first taste of fragment-based drug discovery. We were looking at developing fragments to inhibit the Rad51 BRCA2 interaction for breast cancer. That was my first experience of designing something to be biologically active, going into the lab, actually making that compound, and then testing it. And I thought that whole process was just amazing and never looked back. So yeah, here I am 18 years later doing kind of the same thing, I guess.
With way more advanced tools, I guess, right? We'll get into more of that. That's amazing. Yeah, thank you for sharing that story, Rebecca. And yeah, maybe tell us a little bit more about what your favorite molecule is and why.
Yeah, so I actually think I have loads of favorite molecules, and actually every time like there's a new disclosure, actually maybe then I'm like, oh, that maybe that's my new favorite. So maybe one that really piqued my interest this year is the pan-RAF non-degrading molecular glue from Nested Therapeutics, Nested 628. It's a small molecule that binds to MEK. It glues MEK and RAF together and functionally inhibits the MAPK pathway. So I love this mode of action. I think molecular glues are just fascinating. I think what really gets my interest, though, is that this compound is it's really similar to a predecessor compound. And all they've changed is a pyrimidine to a fluoropyridine. And that gives this molecule a really excellent CNS penetration. So this is now in the clinic and might provide an option for patients who have MAPK mutational CNS metastases. So I'm going to be watching really, really closely to see how well this molecule does.
Yeah, that's very, very fascinating. And I'm very much like you actually with the molecule hopping, if you want. Like I also get like new favorites every time. And yeah, it's great that you bring up little changes can have such a big impact on properties. So it's
Yeah.
really a really fascinating story. Thanks for sharing that with us. And what are you excited? I'm sure, I think I have an idea of what you're going to say, but I'd love to hear it from you. What are you most excited in drug discovery today?
I think there's like two really transformational pieces in my mind that I'm really excited about at the moment. So the first is probably going to be no surprise, the AI side, or the emergence of all these AI methods and tools, AlphaFold being one of those, really transformational ML model that can really change how we do drug design. And the other piece for me is the emergence of all these new modalities. So, molecular glues, degraders, protein correctors. Um, and I think what I'm really excited about is over the next maybe 5 years or so is seeing how maybe these two can come together. So, for example, are we going to be able to use AI to rationally design molecular glues, for example? Because this could open up a whole world of possibilities for some really challenging targets that we've not really been able to make much traction on up until this point.
Yeah, definitely very, very fascinating areas to watch. Totally
Yeah.
with you on those excitements. Yeah, I mean, if you could go back to the beginning of your career and could give yourself some career advice, what would you tell your younger self or our younger audience members that are just starting their careers?
Yeah, I think for me, this one would be to really be intentional about creating some time to read like more broadly, not just about your project or what you're currently working on, but really read across the field. Because if you're anything like me, then you'll be really sort of busy. You've got a to-do list, and you're making slides and you're doing your project work, and that kind of broader reading maybe gets just pushed on the back burner for quite some time. But I think it can be really impactful and it kind of grows you as a scientist. So, yeah, my advice to my younger self would be, go around the lab and ask people what they're reading, which journals they find it interesting, what themes are emerging, what alerts do they set up so that they can keep in touch with everything that's happening. And yeah, be really intentional about creating just a little bit of time every week to do some reading.
I love that. I think that's really great advice. All right. With that, maybe my last question is, what do you like most about Drug Hunter?
So I love all of Drug Hunter. I think it's an amazing platform, and we're super lucky at Isomorphic Labs that we get access to Drug Hunter. It's just like beautifully curated content. My favorite piece at the moment is your patent pages. So where you've gone in, you've curated this list of hundreds of patents. You can see which company these are from, which target. There's some information on the patent itself. And I just imagine that to actually pull this together must take you so many hours. And if I was to do this, it would take weeks, right? So this is a way for me to get it in one glance, the overall trends in the industry, who's working on what are the new hot targets. So I really love that.
Awesome. Yeah, thanks for sharing that. I love that feature too. And yeah, before we get into your talk, I'm gonna show really quickly to the audience, I mean, where they can find that. So yeah, if you go here to the Drug Hunter website, you can find that patent engine here under resources and patent search. And then, basically do exactly what Rebecca said. We're curating patents every month where you can check from different companies, different therapeutic indications, different targets, and find an annotation about those patents here on the left-hand side as well. One other thing that I want to show you and want to just quickly demonstrate is the structure search tool. One of the things that Rebecca is going to mention, I guess, in her presentation is that in the DMTA cycle, the making of drugs, so the synthesis, is going to take a really long time, the longest time potentially of the cycle. So one of the things you want to do is you want to be really deliberate about your design. And one of the tools that you can use for that is the Drug Hunter structure search. So for example, let's say you had a, piperidine and you wanted to, you know, bring in an acetidine instead of that. And also you want to bring in a second group. Let's say you want to, instead of a methyl group, you want a CF3 group. You can put in two completely independent groups here and then search either the FDA approved drug database or the molecule database. So let's here in this example go for the molecule database. With those two different functional groups, we're going to hit search. And what we're going to find is we're going to find case studies. We don't have to sift through multiple different hits that are not drug discovery related and then talk about the synthesis. Those are direct case studies, and they explain kind of why those groups are being used. So let's just click into one case study to exemplify that. So we're going to look into this case study of this CCR4 antagonist here from RAP Therapeutics. And then, scroll down to the place where we're talking about the lead optimization. So what you can see here is they use this acetidine to improve the clearance profile and actually get oral bioavailability. But unfortunately, with this methyl group, and now we're getting into why they use that CF3 group, unfortunately, with this methyl group here, they had a time-dependent CYP inhibition liability. So the proposal was that this is going to be oxidized to an aldehyde and then cause metabolites that inhibit CYP. So here are a few suggestions of groups that you can use instead of this methyl group, for example, that CF3 group, to overcome CYP inhibition that's kind of mitigated in a similar way. And we have, hundreds of more examples for different problems that you can solve in drug discovery with the use of different groups. And with that, browse Drug Hunter, the structure search, as well as the full text search. But with that, I'll hand it over to Rebecca. And Rebecca is going to talk about AI drug design first and how the team at Isomorphics is accelerating drug discovery of new therapeutics. We're all very excited for your talk, Rebecca, and the stage is yours now.
Great, thank you. Just gonna share my screen. Hold on. Are you seeing that?
Yes, it's coming up now. Perfect. Yes.
Awesome. Okay. Okay, so yeah, thank you, Dennis, and thank you to the Drug Hunter team for the invitation to give a flash talk today. As we've kind of touched on, I find the level of innovation across our industry at the moment is just mind-blowing. It's an incredibly exciting time to be a scientist, but it's also really daunting to work in this field where things are evolving so rapidly. So I think these talks are a really great initiative to keep us all connected, to share learnings, and to further foster creativity for those of us working to push the boundaries in the drug design space. So today I'll talk about AI-driven drug design and how at Isomorphic Labs we're approaching this with the mindset to completely reimagine the process, both to accelerate and to improve the probability of success. So I've broken down the talk today into 4 chapters. The first chapter is to take you through how we do drug design at ISO Labs, to give you a glimpse of how we're taking a unique approach. I'll then take you through which predictive models we really need to design drugs in the virtual world. In chapter 3, I'll take you through a data acquisition story based in the fragment space. And finally, in my favorite chapter, we'll talk about the evolution of drug design at ISO Labs. So we're only 3 years old as a company, and actually most of our drug design team have only been here for 2 years. And in that time, our approach to drug design has changed significantly. So that's both regarding the number of models we use, how we use them, and the different strategies that we employ. And so in this final chapter, I'll take you on some of that journey. So when AlphaFold was released by DeepMind, solving this 50-year grand protein folding problem, it was a watershed moment and the proof point that AI capabilities had advanced enough to apply to real-world problems. And this led to the founding of Isomorphic Labs. So as our CEO would say, there's no better application of ML than to advancing human health. So we're not a biotech with AI capabilities bolted on. We've been designed from the ground up to bring AI and discovery science capabilities together to work on these big challenges in drug design. We're currently around 200 people, and we bring varied experience from different tech, biotech, and big pharma companies. And you may have seen earlier this year, we've signed deals with 2 innovative pharma giants, Eli Lilly and Novartis, and we work on a number of very challenging targets together with them. We also have our own internal pipeline, which we are continuing to grow. You may not know, but we're an alphabet company and we have a significant funding envelope. So that really allows us to focus on the science. And finally, and really importantly, we have access to this enormous planet scale compute power, which is really critical to making progress in this space. So you can see our offices on the slide. Our London office is at the top over here, based in King's Cross. And this is where most of us are. And then at the bottom, you can see within the EPFL campus, our Lausanne office. So here you're looking at the drug design process, right from target ID all the way through to marketed drug. And at ISO Labs, we think about 4 key bottlenecks across this process, where we believe that ML is poised to have an impact. So right at the start, we know it's critical to select the right target for the disease and to further build our understanding of disease biology, given that we still see this significant clinical failure due to lack of efficacy. We're still spending, in many cases, years going from hit ID through to advanced leads. So there's a real opportunity here to improve the speed and success of compound design. We often see this quoted 90% failure rate in the clinic. So again, there's a huge potential here to improve clinical success rates. And the fourth bottleneck we think about is one of scalability. So a success for one biological target does not usually generalize to the next target or indication. And here we aim to develop models and approaches which scale across all target classes and all chemical space. So we believe that ML is poised to solve some of these bottlenecks that we face in drug design. And we think that if we can solve some of these, that will trigger a step change in the time to the clinic and the chance of success. So we acknowledge that all 4 of these bottlenecks are of fundamental importance to reimagining this entire process. But today I'm going to focus here on compound design. So here's a question for you all to think about as I walk through this next section. So if you could build an optimal process to design new drugs, what would that look like? What would you need and how would you do it? So here's the classic DMTA cycle that we're all very familiar with. And traditionally, we begin over here in the design phase, perhaps with some chemical matter that we've started with, and we make small changes to improve properties. We then send compounds off to the lab for synthesis, test compounds in assays, analyze the data, and we iterate around this loop until we reach our advanced leads. And the time we spend in this loop varies significantly depending on the nature of the project and many other factors. But according to the Nature paper cited on the slide, the average is around 3.5 years. And this feels about right to me on average from my experience. But if we redraw this cycle scaled to time, it might look something like this, where actually we can do the test, analyze, and design phases quite quickly, and then we often get stuck in this synthesis part. And the way that we view this at ISO Labs is that this design phase is of fundamental importance, because the compounds that you design here determine your success all the way round over here, and how significantly you've pushed your project forward in just one design round. And we want every design round to have a significant impact on project progress so that overall we minimize the number of iterations and the number of compounds made to quickly reach advanced leads. So what about if we could take this experimental real-world design cycle, which takes months, and perform this in the virtual world in a fraction of the time? And in this virtual world, we iterate multiple times before we come back to the experimental real world to verify our compounds. So in the virtual world, the first thing we need to do is generate some chemical matter. And we have a multitude of approaches to do this, ranging from generative chemistry all the way through to an experienced drug designer who's making individual changes or maybe exploring enumeration. And depending on which approach you use, that will influence the number of compounds that you generate in this phase. Once you have some chemical matter in hand, you need to score it somehow against some parameters that you care about for your project. And this is really the critical part of this entire virtual loop. Because if the predictivity of these models is approaching experimental accuracy, then this allows the drug designer to select the molecules which best fulfill their MPO score to take through to the next virtual iteration. And so we continue around this loop until we're unable to make further improvements to the scores. And at that point, we re-enter the experimental real-world cycle, sending designs off for synthesis. And we only take compounds into synthesis once we have a significant improvement on the score versus our starting point. And this is a really key point that I want to emphasize on this slide. This moves us away from the more traditional single or small changes and towards score-based ML-led compound design. And if we think about it, this means we'll always achieve actionable information from synthesis. Either our molecules will have experimental data which aligns with the predicted scores, and we've pushed the project forward significantly, or we've generated data which tells us actually our models are not as predictive as we require, and we need to do something about that. So we consider that this approach to drug design is particularly well suited for fragment-based drug discovery, because it's a way of allowing rapid progress from initial weak binding hits. And there are loads of exciting possibilities for compound design in this virtual world. And if you're interested, I really recommend watching the recent TED Talk by our Chief AI Officer, Max Jarderberg, who's really inspirational in this space. And I also recommend some of the YouTube videos from our CSO, Miles Congreve, who is also really inspirational in this space. So in the way that I've described this to you today, this is really very driven by the chemist who's embedded in this loop and makes those decisions about which compounds to select based on the scores that they see. But the real power here is in the use of AI agents who can carry out these multiple steps and who we can really put to work doing this at scale. So let's now take a deeper look at some of these models that we need to effectively generate and score our compounds in this virtual design world. So firstly, we need ways of generating small molecules. And at ISO, we're building our own generative chemistry models where we can expand or modify a starting molecule or explore an optimal linker to connect two binding ligands together. And you can think of this as essentially exploring the chemical landscape around a starting point as a focused virtual screen in continuous chemical space. And we found that we can increase the value of generative design approaches significantly if we score molecules by synthetic accessibility. So we focus our efforts in the synthetically tractable space and can really start to minimize that huge chunk of the DMTA cycle. Once we've generated some chemical matter, we want to understand rapidly whether we've generated any compounds with improved binding affinity. And one of the really key predictive models we've built here is our novel deep learning binding affinity model. So all this model requires is the ligand SMILES and the amino acid sequence of the protein. And that's your input. And this will then predict you two values as an output. You'll receive a binding probability, which is the probability that your molecule binds to your protein, and a predicted binding affinity, which is a predicted KD value. And the really key point here is that this is a general model. It doesn't have to be conditioned for any particular target, although of course there is potential to do that. So theoretically, you should be able to use this model to make a prediction for any ligand against any protein target. And in benchmarking this general model, we know that it's at least as good as FEP on the the Schrödinger FEP benchmark set, but it's orders of magnitude cheaper to run. So on a single compound level, I can actually get a prediction back in now less than 5 seconds. So it's essentially in real time. I can also then score millions of compounds over the course of maybe a day or a few days. And that allows us to explore really wide chemical spaces during in silico optimization. So on the right side of the slide, you see an example of a blinded prediction study using this model. So this is a GPCR for which we had no experimental structural information. And the graphic shows the AlphaFold predicted structure of the GPCR, with the colored points representing putative binding sites. So the key here was that we were able to correctly identify binding ligands from a test set, and we were able to correctly identify the allosteric pocket shown in green over here as the most likely binding site. And this was later confirmed with the first cryo-EM structure for this GPCR. So as we all know very well, it's not all about potency. Being able to correctly score compounds based on desired ADME and PhysChem is also really important so that we guide design to good property space. And we have a number of in vitro ADME models that we use to score the chemical matter that we're generating. So if I just draw your attention to the graph on the left-hand side of the slide, the blue bars represent some of our early ISO ADME models, and they're compared to the gray bars, which represent an industry benchmark. And lower is better in this case. So we're looking at mean absolute error on the y-axis. So you can see we have models for log D and log P, solubility, permeability, and some in vitro clearance models as well. And we can use these predictive models to set up MPO scores to make decisions on which compounds to progress. Prediction of protein ligand structure is a really critical node for us in the virtual design of molecules. An understanding of the binding mode helps us to define the best strategy for optimization. So, for example, the best vector to explore to probe a specific interaction or to target a specific region of a pocket. And being able to predict a binding mode also then helps us to rationalize our experimental data. So to rapidly predict protein ligand structures, we use our latest in-house structural prediction models. And these build on AlphaFold 3, which was developed by Isomorphic Labs in collaboration with Google DeepMind. And on this slide and in the subsequent 2 slides, I'm just going to talk a little bit about AlphaFold 3, which was published this year, and you can see the citation at the bottom of the slide. So this is a deep learning model that is capable of high accuracy protein ligand structure predictions. And as a medicinal chemist, all I need to run this model is the SMILE string for my ligand and the amino acid sequence for my protein. And I'll be returned with a structural prediction for my protein ligand interaction. A really key differentiator versus docking is that we don't need knowledge of the binding site or a known crystal structure as a template. This model is able to generalize across all target classes, regardless of whether structural information is already known. So the bar chart shown on the slide just compares the percentage success of AlphaFold 3 against Autodoc Vena, which requires a structural input, and RosettaFold on the ligand PoseBusters benchmark set. And you can see AlphaFold 3 far exceeds the performance of other models on these external benchmarks, and it's getting these structural predictions right almost 80% of the time. So I wanted to show you a case study of AlphaFold 3 in action. And I'm sure many of you have been following the incredible story from Revolution Medicines in the discovery of their molecular glues to inhibit the function of KRAS. And so on the slide over here, in the white ribbons and sticks, you can see the ground truth crystal structure of cyclophilin A, KRAS G12C, and the covalent molecular glue in the middle. And overlaid on top in the colored ribbons and sticks is the AlphaFold 3 prediction of this ternary complex. And I think hopefully you'll agree that this prediction is visually outstandingly accurate. And it's all the more impressive given that the cyclophilin A KRAS ternary complex is not in the training set for the model, and the site of covalent attachment is not specified at inference time. And this is actually a really tricky problem, and it's demonstrated by the model stochasticity in this case. So when we supply the model with the same input 250 times, we see this distribution of poses that comes back, indicating that there is some degree of uncertainty in this prediction. So we can use confidence scores to predict the most likely pose. And in this case, you can see that the 3 solutions with the highest confidence over here also have some of the lowest protein ligand pocket RMSD from the ground truth structure. So here you're looking at the AlphaFold 3 APO structure prediction of a protein called TIM3. And upon co-folding with a small molecule published by Steve Fessick and his group, you see the tryptophan side chains reorient to pi stack with the ligand in close agreement with the ground truth structure, which is shown in white. And this is quite a significant side chain movement, which is well predicted in this case. A more optimized ligand with a paramethyl sulfonamide substituent from the same paper binds in a similar way to the protein and is also well predicted by AlphaFold 3. When the sulfonamide is substituted with a 2-imidazole group, the sulfonamide substituents rotate slightly, and again, this is very well predicted by the AlphaFold 3 model. So this example demonstrates how AlphaFold 3 is able to predict the dynamic movement of side chains and ligands and helps the drug designer to understand how their molecule is binding to their protein. So our world-class ML research team continue to further develop this incredible portfolio of both predictive and generative models. And these cutting-edge research models are then available to use directly by our chemists through our drug design platform. So you can see this shown schematically on the slide. So in this box here, this just represents our models. And these are fed by this huge wealth of training data. And the models are then surfaced in our drug design platform over here. So this is really driven by the human drug designer, who can then rapidly generate and score chemical matter to find and optimize ligands according to the specific desirability criteria for their particular target. So we have this incredible suite of models, but we're always striving to improve the accuracy of model performance. And one way to do this is to increase the quantity of training data. So 2.5 years ago, we set out on a data acquisition campaign to generate data in the fragment space. So we've realized over the last few years that the technology we've built is well suited to fragment-based drug design. So firstly, one of the big challenges for fragment-based approaches is the prediction of binding poses, where docking-based methods can often struggle. Well, here we have our latest structural models that allow us to rapidly predict fragment binding modes without the need for crystal structures. And we can make binding pose predictions in just a few minutes. We then have generative chemistry approaches to provide an avenue for rapid fragment expansion. And this is then guided by our binding affinity models to ensure we're exploring the most active chemical space. So to be useful for fragments, we considered that our binding affinity models need to be able to make accurate predictions in the low potency and low molecular weight space. And so we set out 2.5 years ago to generate a dense and square fragment binding data set. And our hypothesis was that this would provide a useful training set to improve the performance of our binding affinity models in the fragment space. And so we partnered with Arcturus, who are based in Oxford in the UK, and we set out to screen 5,420 fragments against 65 kinases to generate this foundational data set to test and train ML models in the fragment space. And we chose kinases for this very first data acquisition project to ensure we were following tractable biology and could make some fast progress. So the first task for us was to choose the 65 kinases for screening. And the key objective here was to select kinases to represent the diversity of the kinome. So we could have done this in a traditional way by selecting representative kinases from the Manning tree, which is clustered by sequence similarity, as shown over here. But we know that the AlphaFold models take into account more than just the linear sequence, and we hypothesized that clustering using this additional information would give us a representation of the kinome from the perspective of the model. So we leveraged the AlphaFold network embeddings to arrive at a cluster set which spanned the kinome. And you can see how these two clustering methods compare in the two pictures, which have both been colored according to canonical kinase subfamily. And you can see that in many ways, the picture is reassuringly similar, with only 9% of clusters containing a mix of different canonical kinase subfamilies. And I'll just draw your attention to some of those families over here. So we made the decision to proceed with the novel clustering using the AlphaFold embeddings, and we chose one kinase to represent each cluster. We also intentionally overrepresented some clusters to build these intra-cluster data sets to see the effect that this would have on model performance on closely related kinases. So the final target representation is shown superimposed on the Manning tree in purple. And our screening strategy for this data acquisition campaign involved taking the 5,420 fragments and screening these against the 65 kinases in a 2-point screen, generating us approximately 700,000 data points. We took the hits forward into concentration response to give us 1,863 active protein ligand pairs. In parallel, we also screened 1,500 kinase inhibitors from a commercially available pre-plated kinase set against the 65 kinases. And we were expecting a really high hit rate here, so we took these directly into concentration response to generate 100,000 data points and 34,000 hits. We then ran a counter screen against all the hits, and we did remove 12 fragments and 27 kinase inhibitors. So to check the reproducibility of the generated data, we took a selection of compounds across both the fragment and kinase inhibitor libraries, spanning a range of potencies, and we retested the selection against the same kinases at a different CRO. And the assays were different, but you can clearly see the correlation between the data. Within the kinase inhibitor library, there were a number of compounds present as salts and free bases, essentially giving us a built-in NF2. And you can see an excellent correlation between the data for the repeat pairs over here. The KDE plot on the right-hand side of the slide shows the variance within the Arcturus dataset in yellow compared to the variance within Kemble shown in red. The Arcturus dataset, as you can see, is much more consistent, exactly as you would expect for a dataset generated in the same lab using the same assay format. The key question that we're now interested to ask is whether we achieve better model performance when training using the lower variance dataset. So then just a little look at the fragment data landscape. So we observed that the fragment data that potencies tended to range from roughly PIC50s of 3.5 to 6.5. And you can see that there's a significant variation in the number of hits per kinase, as shown by the number of colored points. Hit fragments tended to be highly ligand efficient, with ligand efficiencies generally bumping along between 0.3 and 0.6. And we hypothesized that overall, this very weakly active but highly ligand efficient data set is valuable for model training. Surprisingly, the CMGC family tended to identify more hits per kinase than any other kinase subfamily. And this was unexpected. We were expecting that the tyrosine kinase family would be the most promiscuous. So this UMAP plot of all 5,420 fragments is colored by the maximum activity of a fragment against any kinase. And so it's really stark to see that more than 90% of this library is actually never active against any kinase. The 485 unique active fragments tend to cluster over here, and these represent those 1,863 active fragment protein pairs. So having this massive data set in hand is really exciting. There are loads of possible questions to ask and so many different ways to analyze this data. And just one of those questions was to understand the promiscuity of the hits we'd identified. So each kinase on the chart you can see is represented by a single bar, and the bars are colored according to the number of kinases each fragment hits. The total number of fragment hits is shown on the y-axis. So I'll firstly just draw your attention to DIRC1A and CLK1 over here, which both identified the largest number of hits of any kinase, and RIPK1 down here, which also stands out as an outlier. So you can see that the blue color of the bar here represents that the fragments that bound to RIPK1 tended to only bind to RIPK1 and not to any other kinases. So if you were to count the bars on this graph, you'd count only to 64. And that's because we had one kinase for which, surprisingly, we identified no hits at all. We found that 89% of the total active protein ligand pairs are represented by fragments which bind to two or more kinases. Almost 50% of the fragment hits are substructures of known kinase inhibitors, which helps to build confidence. But if we turn this around, 50% of the hits were not substructures of known kinase inhibitors, and these might represent a really rich source of information for model training. So we've now predicted the binding poses for all the fragment protein ligand pairs, and you can see a random sample of 100 of these overlaid on the slide. So those that you're looking at are all predicted to be orthosteric, but we did also identify some compounds predicted to be allosteric. And again, these might represent a rich source of data for training. Since the publication of AlphaFold 3 earlier in 2024, we have further improved the accuracy of our kinase ligand structure models, all the way now up to 89%, allowing us to choose some of the more promising fragments and rapidly expand these using generative approaches in a structure-based manner. So then just to conclude this section, we've generated this really big fragment data set, which is composed of more than 350,000 protein fragment pairs. A huge chunk of this was represented by inactive compounds, but this actually provides really valuable data that's not generally available in the public domain. We've now gone ahead and used this data as a test set, and that's helped us to build confidence that actually our models are already able to predict very well in the low molecular weight space. We've been able to propose binding modes for all fragments using the latest ISO structural models. And then just finally, we've incorporated this data set into our general binding affinity training set. And we saw that this did lead to an overall increase in general binding probability model performance. And I'll just caveat that by saying that we have not yet determined the significance of that increase. So then on to the final chapter. So since I started at ISO 2 years ago, we've been running sandbox projects alongside our main portfolio projects, allowing us to explore different ways to use the models we have in drug design. And these sandbox case studies are a window into ISO capabilities at any particular time. And they really illustrate the journey and the progress made over the past 2 years. So in this very first case study from early 2023, we started from a binding fragment, and we attempted to optimize this fragment using only the AlphaFold structure models. So we replaced an undesirable functional group, and we added an interaction with a water, and we also filled a subpocket. And this was all within one virtual design round. And this gave us a significant improvement in IC50. So this demonstrates that you can optimize a fragment using just AlphaFold alone to guide optimization. However, the gains in potency here were only modest. And amongst these active compounds, we also made a number of inactive compounds. So AlphaFold on its own is clearly not enough to make significant improvements in the efficiency of drug design. In this second example, we combine both the AlphaFold structure prediction and one of our generative chemistry models to optimally link two fragments together. So we start with a literature molecule, which we've deconstructed into a fragment, which is shown here in yellow. We then perform an in silico virtual screen to identify fragments binding at a second site. And you can see this green fragment here, which we'd further optimized using the AlphaFold models to optimize the interactions with the protein. We then used generative chemistry models to link these two fragments together, and we went ahead and synthesized 6 molecules. And really pleasingly, one of these was a potent and novel inhibitor. So in these 2 in silico steps, we've significantly improved IC50, and we've generated some novel chemical matter. But if you really want to improve the efficiency and the pace of drug design, you really need to layer on top a binding affinity prediction. And that's what I'm going to talk about in this final case study. So at the end of 2023, we decided to set a company-wide design challenge. And the remit was to design a selection of molecules against a particular protein target using any approach, but guided by the binding affinity and ADME models. And these were general models, and they've not been fine-tuned or conditioned in any way for this protein. In January 2024, we selected 30 molecules for synthesis, covering a range of chemotypes and a range of predicted binding affinities. So you can see the results in the chart. So you're looking at the experimental PIC50 versus the predicted PIC50. And the points on the graph are colored according to similarity to the genetic cluster training set. So points colored blue represent compounds that look very different to anything that the training set would have, that the model would have seen through training before. And compounds colored yellow or green are compounds that are more similar to molecules in the training set. So firstly, you can see that actually the correlation here between predicted and experimental is very good. For those of you with a keen eye, you'll see that the model is slightly over predicting PIC50. I'll just draw your attention to these points down here at the bottom. So these were compounds that were designed by a very experienced medicinal chemist who was absolutely convinced from a similar target that they'd worked on before that these compounds were going to be novel and very active and were going to push the project forward. And so obviously you can see these compounds have come back inactive. So the model is able to see something here that a very experienced drug designer is unable to see. And you can imagine that without this binding affinity model as a guide, you could easily become stuck trying to optimize in this chemical space rather than finding and prioritizing some of these more active and yet still novel blue-green compounds up here. As mentioned in an earlier slide, we also predict a binding probability. And in this example, you can see that the model is working as almost a completely perfect classifier. So for example, you could draw a line here at 0.95, and you could say, well, I'm not going to make any molecules with a binding probability of less than 0.95 because those molecules are very unlikely to be active. Whereas if I make molecules with a binding probability of greater than 0.95, I'm really going to be enriching for active chemical space. So I'll just finally mention that we also obtained a very good R-squared for our log-D model in this design challenge too, and that made it easier to stick within the desired lipophilicity range when designing compounds. So for one particular design in this case study, we started from a commercially available fragment, which we predicted to make 2 interactions to the protein. And in this one design round, we optimized this to a molecule shown over here, which we predict makes 4 interactions with the protein. The molecular weight is unchanged. This is now a novel compound, not covered by any marquoosh that we could find in SciFinder. The potency has been increased by a log unit, PIC50, and the ligand efficiency and the LLE have also increased. So we now consider that in this one design round, we've been able to obtain a ligand which would be a very good starting point for an optimization campaign against this target. So we've made incredible progress over just a few short years, and we've had really great fun in the process. To do impactful work in this space really takes a multidisciplinary village of really talented people in ML research, engineering, drug discovery, bioinformatics, product, and more, not to mention our incredible operations and people teams. If you just look at the photo on the top right of the slide, these are the 6 original members of the drug design team at Isomorphic Labs. This was back in 2022. Obviously, we're much bigger now. So with that, I'll just like to thank this wonderful team at Isomorphic Labs who've all contributed to the work presented today. And I'd just like to particularly acknowledge those at the bottom of the slide. So Adrian, who led the fragment data acquisition project with me and also conceived and delivered the clustering using the AlphaFold embeddings. Marcos, who did all the fragment data library analysis. Pascal, who's been a real thought partner on this slide deck and also did the KRAS cyclophilin A case study. And Lorenzo and Miguel, who are really 2 superstars from our incredible biology team, who also worked on the fragment data project. So I hope I've been able to give you a small glimpse into how we're reimagining drug design at ISO Labs, and I hope you'll continue to follow us on our journey. Many thanks for your attention, and of course, I'm happy to do my best with any of your questions.
Awesome. Yeah, thank you so much, Rebecca. That was an incredible talk. And I mean, it's really cool to see how you can accelerate the discovery of starting points with that last example that you gave. So really, really, I think game-changing technology. And yeah, we have a lot of questions already, so let's get right to it. Hopefully, we can address most of them. If not, we might later come back in writing. The first question here is from Alexei from Schrödinger, and he's wondering what was used as industry benchmarks for the ATME models that were compared with the ISOLAB models.
Yeah, so it's a great question. We're always benchmarking all of our models against any industry benchmarks that we can get our hands on. Unfortunately, we don't actually disclose what they are. That's why they were blinded in the slide. But we are very careful to benchmark everything that we're developing against as many industry benchmarks as we can get our hands on.
Sounds good. Yeah, thank you. And then there's another question here from it's about it's from Michael. It's about like the reverse mode. So is it actually possible, like, to see an active ligand from a phenotypic screening and find possible targets with this technology? And how accurate would something like that be? So rather than basically, finding a ligand for a protein, finding a protein for a ligand.
Yeah, that's a really interesting question. Well, obviously, the way that we've set up our models, it's very much that we can model protein ligand interactions. So we're very focused in screening that way around. One of Demis's visions is that eventually we'll move towards building this virtual cell. And so I imagine if we can achieve something like that, then being able to deconvolute a phenotypic screen should be something that would be within the remit of that kind of piece of work.
Cool. Yeah, and then we have another theme emerging that's asking about molecular weight or molecular size limitations. So people are noticing that mainly molecules, like small molecule therapeutics, are being discovered in the AI space. And maybe could you comment on whether there's a molecular weight limit and whether maybe there's some ideas for engineering or designing therapeutic proteins, so somewhat larger molecules with that approach.
Yeah, so we have been very focused in the small molecule arena up until this point. But we are currently hiring a world-class biologics team, as people may have seen from LinkedIn. So this is, something that we're excited about, and we'll continue to grow in that space too.
Awesome. And then another question from an anonymous attendee here is, do you think crystallographic fragment screening will become obsolete with these AI models?
I mean, yeah, that's a great question. Possibly. I mean, I would never want to rule out anything at this point. I mean, we can predict, with reasonable accuracy most of the time, a protein ligand binding pose. And so actually, and we can do that really quickly as well. So, there'd be less value in then going out and generating, doing a crystallographic fragment screen. Of course, we'd want to validate binding poses for new targets. So that doesn't mean that crystallography is obsolete by any means at this point, but certainly I can see that you would quickly determine a binding mode using the AlphaFold or the structural models that we have. And you'd be much less likely to screen using crystallographic fragment screening. Yeah.
Yeah, that makes total sense. And we have a question here from Niels. He's wondering, with modern generative chemistry approaches, you can quickly sample large portions of the chemical space, but at the same time, reward hacking has been observed several times. How do you ensure your methods are robust and don't just exploit glitches in the complex MPO scores we have to use?
Yeah, that's a really good point. And I think at the moment, we very much see that the human needs to be a bit in the loop here to keep an eye on this and to make sure that, you don't have models which are just putting on like 20 adamantyl groups to optimize binding affinity, right? This is why it's really important that, you still have experienced drug designers who are driving this system at this point. And that you make sure you set your MPO score up properly so that the binding affinity model isn't just completely taking over and increasing molecular weights up to some completely unacceptable level. That you're really balancing all properties across phys chem, ADME, and binding affinity.
Sounds good. We have another question here from Brad. How do you allow for serendipity in drug discovery? Many times, key advances happen from unexpected effects.
Well, that kind of serendipity is still there, right? Because at the end of the day, we're making predictions, but we don't know whether those results are going to to play out in the real world. And sometimes, these predictions, you think, oh, maybe this is quite speculative. And if this works, this would be great, but maybe, maybe it's something that you're not completely sure about. So I think that the door is still open for these serendipitous discoveries. But, I think what we're trying to do is to make sure that, if you like, we're fishing in the pool with the most fish. So really enriching what we're doing for, the chemical space where we're most likely to find the best chemical matter.
Makes sense. And we had kind of another theme emerging in the chat as well as in the Q&A. And that was one thing was how are the algorithms and AI models accounting for water molecules in the binding? And also how are they accounting for like molecular dynamics, for example, closed and open stages. We talked a little bit about molecular glues in the beginning. Ion channels oftentimes have closed and open stages. So is there any way that the AI models contribute or like, take that into consideration?
Yeah, so the models seem to have this sort of, I guess, implicit understanding of where waters might be. So they kind of take account those into account. In terms of kind of making predictions against different conformational states of proteins, this is something that we're super interested in, and I think this is like probably the most important next step. So it's something that we'll be working on over the coming years.
Yeah, sounds good. And then let me see. How did you build your models for ADME predictions? Did you use internal or external databases?
So we'll use data from wherever we can get our hands on it, but so that would include public sources of data. But we see data generation, really highly value data generation here, and we do invest in that. So we do invest in our own proprietary data sets as well, as you saw with the data set that we've generated as part of the Arcturus project. We see that as a really important part of building consistent data to train models.
Yeah, sounds good. Then yeah, we're coming almost up on time. Like maybe a few more questions. One question here from Cheng was, could you talk a little bit more about the correlation that you found about post prediction and binding affinity prediction?
So do you mean as in, do the two always correlate together?
Right. Like, yeah, if I understand the question right, for post prediction, Cheng is saying something like RMSE, and for binding affinity, something like PKI.
Right. Okay. Yeah, I mean, I guess these are two separate models, right? The one is a binding affinity prediction, and one is the AlphaFold models that can predict a structure. So there's not necessarily a correlation between the two. You have to use your judgment as a medicinal chemist as to how you interpret the data that comes from both of those models.
And maybe as one last question to wrap it all up, how are you typically taking negative predictions into account? Let's say, you get a molecule that is predicted to be negative. Would you still be making this, or like how do you deal with those types of negative predictions?
Well, yeah, it's a great question because I guess early on, like maybe a year ago or more, as a team, we had less confidence in the models. Obviously, we hadn't used them. So we would make lots of compounds that were predicted to be negative because we were like, well, using our medicinal chemistry knowledge, these are definitely going to be active. But, after getting back a lot of negative data, I think we're really learning that actually we really should be using the models. And the models we have really do significantly enrich for active compounds, and we're seeing it across all the projects that we're working on. So I think we're making less and less of those less well predicted compounds and building that confidence in using the models.
All right. Yeah, thank you so much, Rebecca. I mean, this was incredible. I think you gave a phenomenal presentation. Thanks for sticking around to answer this round robin questions. And we have so many more questions here from people. There's just really high interest in that area. And I think you've done a phenomenal job to answer the audience questions. So I want to thank you for your time today and for presenting this super exciting topic to the audience. I think you've inspired many audience members, including myself, to like keep a close eye on this area. I want to thank our audience as well for being engaged as always and asking that many insightful questions. So really thank you for being engaged. This is what the Drug Hunter community is all about. And yeah, with that, we close the seminar for today, and I encourage everybody to sign up for our next coffee chat, which will be next week on Friday. And we will be talking. Rebecca today answered the question about her favorite molecule, which I think had a phenomenal answer with this nested molecule that she highlighted. We're going to talk about the favorite molecules of 2024 off our Drug Hunter team. So encourage you to write in your favorite molecules and molecules you want to see us cover. And you're going to hear some of the favorite stories that the team has. So until then, and Happy New Year and happy holidays to everybody. Thank you, Rebecca, again. It was phenomenal talk.
Thank you very much.
Bye now.